RNA editing is a widespread process in many life forms, but it is particularly prevalent in cephalopods, a group of mollusks that includes octopus, squid, and cuttlefish. Though the full extent of how RNA editing influences the characteristics and behavior of cephalopods is not entirely known, there are more and more links between the particular traits of cephalopods and their levels of RNA editing being discovered. So what is RNA editing? RNA editing is when RNA gets edited or changed after it has been transcribed from the DNA, but before it is translated into a protein. RNA editing has two main mechanisms of accomplishing this, being substitution editing and insertion/deletion editing.
Here is an example of substitution RNA editing. In this case, CDU. What is happening in this instance is an enzyme will catalyze a chemical reaction at a specific site on the RNA that will result in a changing of the single nucleotide, much like a point mutation. In this case, it's cytosine to an uracil. This results in what would be coding for an amino acid glutamine being transported into a stop codon, which results in the translation of an entirely different protein. Thus, due to RNA editing, the body is able to both produce a protein that helps transport cholesterol through your blood and a protein that removes lipids from the intestine from the same bit of DNA. Here is an example of insertion and deletion editing. Unlike substitution editing, insertion and deletion editing uses a guide RNA to make the changes. The guide RNA is lined up with a messenger RNA and bonds to it as best as it can. Because these strands of RNA do not directly correspond with each other, bulges of unmatched nucleotides will form out of the unmatched nucleotides. To fix this, an enzyme opens up the messenger RNA and allows for bulging nucleotides on its end to be taken out and new nucleotides to be added in to pair with the corresponding unmatched nucleotides on the guide RNA. These edits are especially significant because they can allow for frame shifts in the RNA sequence. RNA editing has a lot of benefits to it. It allows for a single gene to encode for many different proteins, allowing for an organism to have a much larger proteome without increasing the size of its genome. Because RNA editing is only temporary, it is ideal for making quick adaptations for a temporary environment because an organism will be able to quickly discard the adaptations once they become unfavorable, unlike permanent evolution. A known example of this is temperature adaptations, where RNA editing will occur to allow an organism to adjust to the current temperature by editing their RNA. An example of this in cephalopods is how a tropical and Arctic species of octopus each has a gene encoding for a voltage-gated potassium channel that differs from each other only at 4 nucleotides that have no effect on the electrical properties of the channel. However, because messenger RNA of the gene is so extensively edited, each has electrical properties that are adjusted to allow the channel to properly function in the temperature of each species' respective habitat. Octopuses are also known to flourish while other species struggle during turbulent times in the ecosystem, their ability to quickly adjust to temperature likely giving them an edge in times of rapid change. RNA editing in cephalopods differs heavily from in mammals in several different ways. For one, RNA editing in mammals primarily occurs in genetic repeats, repeated sequences of DNA and RNA. While RNA editing in cephalopods occurs in genetic repeats at similar rates to humans, it accounts for only 21 to 38% of RNA editing in octopuses, while accounting for over 95% of RNA editing in primates. Also, while only a small amount of instances of RNA editing in mammals result in the creation of a new protein, a very significant amount of RNA editing instances in octopus produce new protein at a rate of 11% to 13% in neural tissues. Most agRNA editing sites, usually where AIRNA editing is performed, are shared between the common octopus and the two-spot octopus at around 60% being conserved, and even the distant species of octopus share many agRNA editing sites. Furthermore, there is also tremendous overlap between cuttlefish and squid and agRNA editing sites at roughly 35% of editing sites being shared between the two, and a smaller but still substantial number of shared sites between octopus and squid, as well as cuttlefish and octopus at rates at about 10 to 12% depending on the species of octopus. In contrast, only 1 to 2% of human agRNA editing sites are shared by mice, and a similar number are shared between species of flies that diverged evolutionary at a later time period than squid and cuttlefish did, which is significant as there is a positive correlation between time and evolutionary divergence and shared agRNA editing sites among different species. Furthermore, these shared sites are utilized at similar rates across the different species, with the similarity of usage being heavily correlated with how close the species were evolutionary, and there are 1,146 editing sites shared by octopus, squid, and cuttlefish. Interestingly enough, While mammals tend to do less RNA editing in sites that have been evolutionarily conserved and are shared across different species, cephalopods tend to have the most RNA editing at their highly conserved and frequently shared editing sites. All this points to heavy agRNA editing being developed by squaloids before evolutionary divergence of different species, which gave them a shared base of the aforementioned 1,146 RNA editing sites, and then RNA editing sites being developed to be tailored to the individual roles of the diverging species of colloids. The affinity cephalopods have for RNA editing also fits in with how, generally speaking, they have evolutionarily been geared towards the short term instead of the long term. Lacking specialization towards a particular niche, the octopus's structuralist body, ability to camouflage itself, and ability to escape by creating a cloud of ink, make it skilled at hunting for food out in the open, as well as make it able to hide and live almost anywhere. Most vertebrates camouflage themselves by adjusting their positioning in only certain areas in order to fit their environment. But octopuses can directly adapt to the coloration texture of the environment that currently surrounds them, further showing their inclination towards temporary adaptation rather than permanent adjustment. This unique ability to camouflage is likely due to RNA editing, as large amounts of RNA editing has been found to occur in genes that code for reflectin proteins, which play a role in the camouflage of cephalopods by allowing them to produce iridescent colors. Because of the risk associated with not having a shell and being forced to hunt out in the open, octopuses have also evolved to have very short lifespans to reproduce very quickly in large numbers. Since the presence of RNA editing sites restrict genome and thus slow natural evolution, the octopus, as well as other cephalopods, has likely evolved to halt its own evolution in order to stop it from specializing and losing its short-lived, highly adaptable life cycle. Furthermore, the ability to edit the proteins in its body on the fly likely helps to enable its fast-paced lifestyle, and quick adjustment is key. RNA editing slowing down evolution would also explain how many different species of octopus were so similar they were recognized collectively as Octopus vulgaris despite branching off evolutionary from each other millions of years ago. With RNA editing sites as common as they are in the octopus, the genome would be rigid to the point where the opportunity for evolution would be so limited that the different species of octopus could only slightly alter their physical structure through evolution, despite living in different habitats for very long periods of time.